one. Welcome. Hello, everyone. You've reached the CyVerse Focus Forum webinar series. I'm Tina Lee, CyVerse's User Engagement Officer. Our presentation today is by Kristopher Kieft on Vibrant, a tool for automated recovery and annotation of microbial viruses from genomic sequences. Chris is a doctoral student at University of Wisconsin-Madison in the Department of Bacteriology in Karthik Anantharaman's lab. And as a Goldwater Scholarship winner back in high school, Chris said his career goal was to, quote, get a PhD in virology and conduct research on pathogenic viruses, specifically molecular mechanics of infection. Well, he's obviously on his way, and he knew well on several years ago what he wanted to do, so we're happy to have him here today. For those unfamiliar with CyVerse, we are an NSF-funded cyber infrastructure project, and today's free webinar is part of a series that we've designed to fulfill part of our mission to train scientists like you on how to use CyVerse's computational resources. I'll quickly take care of housekeeping and then we'll start the webinar. Chris's presentation today is roughly 30 minutes with time for Q&A at the end. Open your chat window if you have not yet and type your questions there for Chris and we'll address those after he's done. Materials from today's webinar, such as Chris's slides and the video recording, will be posted on our website events for this webinar. CyVerse just concluded a successful week with about 40 eager participants who wanted to learn about foundational open science skills. We'll do that again next year, so stay tuned. And then starting March 10th, we'll be hosting our third container camp, which is all about containerizing your analyses for improved scientific reproducibility. And it helps you greatly to escape from what we fondly call dependency hell. So please visit our website again for more information on this training and to register for Container Camp. But without further ado, I'm pleased to introduce Chris, creator of the tool Vibrant. Hi, Chris.
Hi, thank you for the introduction. So I have a lot to say about this project, and hopefully I can address everything that the people watching are interested in. And if there's anything else you want me to address, I can definitely talk about that. But I'll be talking about the tool Vibrant that I developed in the last year or so, and how this can be useful for your research. So quick outline of what I'm going to go over. So first, I'll start with an introduction to microbial viruses. If you're unfamiliar with viruses that infect microbes or viruses as a whole, hopefully just a couple of slides will get a good intro to that. And then I'll hopefully convince you that Vibrant is a tool that both performs well and can be useful in your research. So I have the acronym there. Vibrant is virus identification by iterative annotation. So I'll tell you about virus identification, and I'll talk to you about how annotation helps with that. And then at the end, I'll talk about how CyVerse can really help you with running Vibrant in these analyses, because using Vibrant on a laptop might not be able to perform what you want it to do. So this is a quick screenshot of the CyVerse discovery environment. If you open up the apps page and type in Vibrant, currently Vibrant is under some maintenance, so I actually don't think it's available right now. The newest version is Vibrant 1.2.0, and when that does come online, hopefully in the next few days, next week, Vibrant 1.2.0 is the version you'll be looking for. All right, so I'll come back to CyVerse at the end, but some key points that I'll talk about is viruses that infect prokaryotes. So I'll specifically be talking about bacteria and archaea and the viruses or bacterial phages that infect these organisms. You can ask questions about eukaryotic viruses. I can talk about them at the end. My previous research was on eukaryotic viruses, so hopefully I can implement some ideas into any questions that you have, but I'm specifically talking about mainly bacteriophages. And then the reason I've developed Vibrant is because viruses are an important factor in microbial communities. So identifying the viruses and identifying what they're doing can be very important, because viruses can augment nutrient cycling. They can play a role in human health and disease or maintenance of disease. And teasing this apart is likely going to be very important for modeling or predictors, or understanding the general ecology of your system. And the other point of developing Vibrant is that we don't have, I'll say, good bioinformatic tools for identifying viruses. And this is actually mainly because identifying viruses and microbial sequences is a relatively new topic, and there's relatively few people working on this. So continuously developing new tools to either outperform existing tools or even supplement existing tools will be very important for future research. And of course, I'll be talking about Vibrant and how CyVerse can help. So here's my couple slides on introducing viruses and what they are and what they do in the environment very, very generally. So in the environment, you'll have a bacteria, which I have this little purple thing, and the bacterium can be infected by a virus. The virus will be floating along, attached to that bacterium, and it inject its genome right inside the bacterium. And that's generally how it works. So what you get is just the bacterium and the viral genome. So for the most part, with bacterial viruses, the genome of the virus is all that really matters. The genome encodes all the proteins, all the genes, all the functions that are going to happen during infection. And that includes things like host modulation, and I'll talk a little bit later about metabolic genes and other genes that will build new viral particles. So that genome will encode all those functions, encode for the ability to build new viral particles, and at the end of infection, through certain mechanics, the virus will lyse open the bacterium cell. And this last part is actually really important, because not only are the viruses coming out of the cell and can spread throughout a community, but that lysis event releases everything that was inside of the cell. So all those metabolites, the secondary compounds, toxic or non-toxic compounds, proteins for the surrounding community to eat or whatnot, is all out in the environment for interaction with other microbes. There's another part of viral life cycles that I will sort of get into, but this is when the viral genome is injected into the host, and then it integrates into the host genome. So my little diagram here is the host genome in blue, and then the viral genome in red just integrates itself right into the host. And that genome can persist over time, eventually come out of the genome, and move through this same cycle. So what I want to point out is that when we're looking for viral sequences in a metagenome or any data set that you have, we're looking for both these free virions, these free viruses, and grabbing those sequences, but also sequences that are actually integrated into the host genome, because those are still important for community function. And like I said, this lysis event is very important to the food web. So I'll show you why that's true here. So here's the start of a food web in general. You have your autotrophic organisms, which are doing things like photosynthesis or other types of autotrophy, but they start at the bottom of the food web, like grass or trees. And then you have heterotrophs, which are eating organic matter and growing off of basically carbon. And these organisms are constantly turning into organic matter, being taken up as organic matter, and recycling over and over in the food web. But this process isn't unchecked. So there's constantly viruses infecting autotrophs, there's constantly viruses infecting heterotrophs, and these viruses are manipulating how those organisms are turned into organic matter, how they uptake organic matter, and viruses are thought to be very central to the recycling of nutrients in the environment. And again, that's mostly because of that lytic event where the host bursts open. And why this is important is because this happens on a huge scale. So an estimated 20 to 40% of all microbes, and this metric is from the oceans, but 20 to 40% of microbes are killed every single day. So there's constantly a huge turnover of a third of all microbes every single day that is facilitated by viral infection. So just to give you a representation of what that looks like, this is a picture I took from the lab of, I have a bacterium, this is Lactococcus growing in a test tube, and you can't see through the test tube because there's so many bacteria. And I added a virus that infects this bacteria, and over time, over the course of about 3 to 4 hours, the bacteria is completely lysed and the tube is almost clear because the bacteria is just gone. So this is the power of what viruses are doing in the environment and the human gut and basically anywhere on Earth. All right, so that's my background on viruses, and then transitioning into the data that Vibrant takes in. So Vibrant was built for metagenomes, so metagenomic data, but if you have single amplified genomes, if you have isolate genomes or any kind of genomic data, if it's a single sequence or hundreds of thousands of sequences, Vibrant can use that. But how this is applied to metagenomics and why Vibrant is necessary is because we need to pull viral sequences out of the rest of the community, separate them from bacteria. So in what I have pictured here is a standard microbial community. It's mostly bacteria here. And in this community are tons and tons of viruses. Interestingly, in most environments, viruses outnumber the number of bacteria. So viruses are super abundant. And one thing we can do is take that community and extract all of the genomes and just have them all mixed together in a pool of genomes. And mainly through Illumina sequencing, but other types of sequencing, you would fragment those genomes into a big mix of fragments, sequence them, and then try to reassemble them back together. And what you get is a ton of intermixed genomes from bacteria and viruses. Some of them are complete, some of them are not complete, and it's just a big mix. And then you have to separate them. So what's a microbe, what's a virus, and what's neither of those? And the reason this is difficult is because this is what the data looks like. It's just a ton of A's, T's, C's, and G's, and all A's look like A's and all C's look like C's. And there's not a good way to differentiate which A's, T's, C's, and G's belong to a microbe and which ones belong to a virus. So what's the best way to do this? How do you start and how do you go about doing this? So first of all, looking at protein annotations manually is really the best way to do it. And there's obvious problems with this. If you have hundreds of thousands of sequences, you can't manually look at all of them. And if you don't have some type of background knowledge on what to look for, that also makes it difficult. But if you were to manually look at a genome and decide if it's a virus or not, my suggestion, what Vibrant implements, which I'll get to, is annotation with 3 different databases. And if you don't need to pay attention to the details really, but just very generally, I use the VOG database, VOG, and as you can see, that's a virus orthologous group. So as you can imagine, VOG annotates viruses. I also have the PFAM database, and this is a collection of protein families, and it includes both viruses and both viral and bacterial annotations, but it's very heavily weighted towards bacteria. for some reasons I can get into if you have questions. And then the KEGG database, which is a collection of metabolic genes. And again, this has very, very few viral annotations and mostly bacterial and other annotations. So just to recap, we have VOG, viral, PFAM, a little bit of both, but mostly bacterial, and then KEGG, mostly bacterial. So if you were to use these annotations to differentiate viruses from microbes, you would say microbes have more KEGG and PFAM annotations and less VOG, and viruses have more VOG and less KEGG and PFAM. Pretty straightforward. So if you were to do this, it would look something like this. So let's take this genome here. Each of these gray arrows represents a gene, a predicted protein. And we can annotate them with VOG, PFAM, and KEGG in red, blue, and yellow. So if this genome belongs to a virus, we would assume it gets annotated with red. If it belongs to a microbe or a bacteria, we would assume it gets annotated with blue and yellow. And we can differentiate them just based on the ratio, or if you look at the title, the abundance of annotations. So what happens when you get this genome? You'd say, well, 8 out of 8 of the annotations are PFAM, so this must be a bacteria. So then you look at the annotation information, and you see that the information tells you that this looks a lot like a virus. So you have viral integrases, you have major spike protein, which is a hallmark of a viral protein, and so on. You have lots of viral proteins. So the idea that abundance can tell you the source of the genome kind of fails in this circumstance. So you have to use both abundance of VOG, PFAM, and KEGG annotations and the information that they give you. So if you want to automate what the information says, how do you do that? So this is basically what I just said. The rule of abundance doesn't really hold up. So the update you have to do to look for this is to say that viruses have more virus-like annotations and microbes have more microbe-like annotations. Pretty straightforward. So what Vibrant implements, and again, what I'll get to, is V-scores. So this is just a random name that I made up, V-score, and this is a number that I give to every single annotation that Vibrant uses, and it just gives that annotation a number. High numbers mean viral, low numbers mean microbial. So super quick the way this looks. These two annotations have V-scores of 10. They look like viral genes. And then something like this has a V-score of 0 because it looks like a microbial gene. So you can look at both the number of annotations and what the annotations are based on V-scores to differentiate viral from non-viral annotations. And I think this is one of the biggest positives of the way Vibrant works. So moving on and getting into Vibrant and how well it performs. So what I wanted to share is why this is a difficult task and why building a program like this isn't easy. So if we look at some made up data and we plot maybe KEGG annotations to VOG annotations, and we say that bacteria are in blue and viruses are in orange or red or whatever color that is. And if we wanted to separate them, oh, sorry, getting ahead of myself. If we wanted to separate them, we would maybe draw a line here and say that everything below the line is viral and everything above the line is microbial. So we can gauge how well that estimation was based on a couple metrics like recall and specificity. So recall is just the true positive rate. So the viruses that we got below the line would be our recall. And then we can measure how many bacteria we called as viruses. That would be our false positive rate. So below the line, we also have blue dots, which would be false positives. So there's different ways of dividing the data and then gauging how well that division actually worked. So I'll show you another example quick here. And this would be not drawing just a linear line, but trying to maximize the way that you separate data. And this maximization, to do this, I implemented machine learning, which again, I'll get to later. So how about data here? How would you build a program to maximize recall and minimize false positive rates? So you could draw a line here, and you would have very high specificity. So there's only one blue dot called as a virus below the line. But you also have low recall because you missed a lot of the orange dots up top. You could also draw a line here, and you would have low specificity because you called a lot of bacteria as viruses. But you did have high recall. You grabbed pretty much all the viruses. But what's likely the best option is to maximize recall and minimize false positive rates, or maximize specificity. So some type of wavy-ish line that grabs as many orange dots as possible and as few blue dots as possible. And that's what Vibrant does. So this is just a PCA plot from Vibrant's output, showing you what real data looks like. I have viruses in green, plasmids in red, and bacteria and archaea in blue. And Vibrant wants to be grabbing all of these green dots and calling them viral, not grabbing any of these red dots or any of these blue dots as viral, get rid of them. And what Vibrant really works at, and at the core of the program, is what to do with this information and things like these red dots that show up in the green area. How do you separate those sections to make sure that you aren't grabbing any bacteria and calling them viruses? So the solution to doing all of this with annotation and separating bacteria and viruses, it all needs to come together in automated software, and then that would be Vibrant. So I have the workflow on the right, the way Vibrant works, more or less straightforward what's shown there. I don't need to go over it, but if you just look from top to bottom, you'll see where CAG, PFAM, and VOG show up. And then where the identification of viral scaffold shows up, and where machine learning is implemented. But Vibrant uses both the abundance of annotations, so generally off the idea that less CAG annotations is good, more VOG annotations is good, and it implements those V-scores to look at information. So it uses neural network supervised machine learning to parse all that data apart. And then I also implement some validation cutoffs at the end. And that's so anything that the machine learning model may have missed, I have some curation steps that can basically smooth out those creases. And then at the end, it has a lot of different automated steps of providing the user information about the annotations, metabolic analysis. If you're familiar with auxiliary metabolic genes, it highlights those. And then what I think is cool that I put in was this completeness estimation metric. So Vibrant can tell you if the viral scaffold that it identified is of high quality, medium quality, and low quality. And this can help you in your analyses to say, maybe you want all the viruses that are high quality. And you can say, I have this virus that's low quality. It's very likely a virus, but it is just a piece of a virus. So I have some comparisons. So this is just a slide providing the softwares that I compared Vibrant to, because I can tell you how great it is, but I need to show you that it works. So you can be convinced that using Vibrant or supplementing with Vibrant can be a great idea. So I'll be comparing Vibrant to VirSorter, VirFinder, and Marvel, which are all virus prediction softwares that exist. So what I have here is a graph that I'll show you about the performance of Vibrant. And again, on the right, if you look at the key, you'll see that Vibrant in green is compared to VirSorter, VirFinder, and Marvel. I won't get into why there's two VirSorters and two VirFinders, but it's basically you can set lower or higher scoring thresholds to change how many viruses you get. But Vibrant just gives you the viruses, and those are all of the confident predictions. So what I'll show you first is recall. So this is the number of viruses or the proportion of viruses out of the total that Vibrant can identify. So if you give Vibrant 100 viral genomes or 100 pieces of viruses, how many does it actually find? And that number is about 98% from the data set that I tested, which isn't too far off from some of your sorter's predictions or Marvel's predictions, but that difference of 4 or 5 or 10% can be a big difference for metagenomes. The next metric is accuracy. So if you predict a ton of viruses, are you actually predicting accurately? So this is the minimization of false positives. And again, Vibrant is accurate. Precision is another relative metric of that, same with specificity. F1 score on the far left takes into account both recall and specificity, so it's kind of a holistic measurement. Same with MCC, and again, Vibrant performs quite well in comparison to the other softwares out there. I have this slide, which I don't need to go over for time's sake, but this is showing just recall, so the amount of viruses identified across, on the x-axis, a ton of different environments. So you can see that throughout whatever environment that you may have for your data, Vibrant can perform well in the data environment that you have, which includes human, marine, freshwater, etc. And again, I won't talk about these, but these are some additional analyses, such as RNA viruses, archaeal viruses, really short fragments, and eukaryotic contamination. You can see those metrics later if you're interested. I mentioned at the beginning that Vibrant identifies those integrated viruses called proviruses, and this is really important to basically grabbing all the viruses possible. So if you have an entire bacterial genome and in just one little piece of that there's a virus, how do you find it? How do you grab just that viral piece? And I'll go over quickly how Vibrant does this. So I have different types of annotations labeled in different colors, but what's important that I want to show you is that in the pinkish purple colors are annotations with low V-scores. So the information that those annotations provided said that those likely belong to bacteria. And the rest of the colored annotations don't have low V-scores. So what Vibrant does is it identifies those regions that have low V-scores and just cuts the scaffold right there and removes those regions. And what you get is that middle region that may or may not be a virus. Vibrant will then trim down that cutout region to the first viral-like annotation, and then that trimmed out cut scaffold is put in with the rest of the free scaffolds. So after Vibrant cuts out a possible provirus, it analyzes it just like all the other scaffolds that you put in. So you get this nice trimmed version of a possible provirus. And this is just a validation that I did showing that what I'm trimming out of bacterial scaffolds based on manual verification that you don't have to worry about, it's just all implemented in Vibrant, is accurate. And here again is more data that I don't need to go over, showing the comparison of Vibrant's provirus prediction to 3 different programs that can also predict proviruses, including VirSorter on the far right. if you've done provirus prediction, you may have heard of FASTER, which is one of the most widely used provirus predictors, but FASTER isn't really made for metagenomes. Like I said, Vibrant will also give you quality or completeness estimations of the predictions. So is that virus of high quality? Is it low quality? And this is some benchmarking that I did to show that if you have a viral genome that's 100% complete, it's more likely to be called high quality. And if you have a genome that's 10% complete, it's more likely to be called low quality. And this works no matter what viral genome you have or what's encoded on the genome. It's agnostic to what you have. So why should you use Vibrant, basically, is the moral of this slide here. And I'm getting towards the end. So if you have your metagenome, why is using Vibrant going to outperform the other programs? Oh, sorry, I'm actually on the wrong slide. So let me back up and say that Vibrant also does metabolic analysis. So it grabs the metabolic genes on viral genomes and automatically gives outputs a analysis of the metabolic capabilities of the viruses. So here's a comparison of the human gut virome or a soil virome, and how the metabolic capabilities of viruses between the environments can be different, and Vibrant can help you figure that out. So going back to what I was saying, why should you use Vibrant? So what I have here is a relatively messy table, but what it includes is 6 different metagenomes. So these are just whole community metagenomes, and at the bottom there's a virome. I have how many sequences were in that metagenome, and then I also have how many sequences Vibrant's actually looking at. So I didn't mention it, but Vibrant can only identify viruses that are at least 4 open reading frames long. So keep in mind that Vibrant only looks at a subset of the metagenome, but it still works great. So within each of these, I'm going to show you comparisons between Vibrant, VirFinder, and VirSorter in terms of how many viruses were identified, what's the total length of all the viruses combined, and how many viral proteins were identified. So let's start with the top one. So I have Vibrant identified 527 viruses from this metagenome. VirFinder identified more. It identified 604. But if you look at the other metrics, Vibrant identified 3 times more total viral sequence. So all the sequences were basically longer, and identified 3.5 times more viral proteins. Because as opposed to VirFinder, Vibrant is identifying genomes that are longer, of higher quality, and more amenable to downstream analyses. And again, this is for your own benefit to look at the rest of the data. Vibrant may not identify as many viruses as other softwares, but it gives you more information, identifies more total viral sequence, it identifies longer viruses and more viral proteins. And then basically final slide, why use CyVerse? Why is CyVerse useful? So I have some run times of how long Vibrant takes. So this top one, I have a file that comes on the GitHub page. It's 200 kilobytes. It's tiny, just has 10 scaffolds, and it takes a couple minutes on my laptop. It's fast. But let's look at some other stuff. So again, this was ran on my laptop. These are relatively small data sets, not that many viruses, and they take, I don't know, a few minutes. But let's look at the metagenome. So this freshwater lake metagenome, it's 254 megabytes, about 1/4 of a gigabyte. It has 80,000 sequences. I ran this on our lab's Linux server, so it's pretty powerful. And if you look at the far right, I have number of parallel runs. So this is basically how many threads was used. So your laptop will probably run no more than 5 max, and you're maxing out your laptop. So I ran this data set on 1, 5, and 15 threads, and it took 548 minutes down to 44 minutes. So with basically 3 full laptops, it still took 44 minutes. So not that usable in your laptop, so you'll likely need to upload it to CyVerse if you don't have access to another server. Workflow of how to use Vibrant. It takes in either metagenomic scaffolds, or you can actually input your own proteins if you've already predicted proteins. I have a little tip here that if you run Metaspades or another assembly software on CyVerse, you can take those assembled contigs and put them right into Vibrant. And I have a link of why I said Metaspades specifically. Here's a bottom left is a window of what you'll see when you try to run Vibrant. It'll ask you the format of the input. Are those nucleotides, something straight out of your assembly, or are they proteins that maybe you predicted with Prodigal? The number of parallel runs, I would suggest, I believe CyVerse more or less will let you go up to 16. So I would say, depending on how many runs you're doing, something between 1 and 16 is good, a higher number for a metagenome. You can specify what's the shortest length of all the scaffolds that you want to identify based on nucleotides or number of proteins. Vibrant can do either. Defaults are 1004. And then there's some other different options that you can check out the GitHub page to see those. These are just the different outputs from Vibrant. So it gives you basically a ton of different information. It separates out all your viruses, lytic, lysogenic. It gives you metabolic analyses, all the annotations, tells you which sequences are circular or complete. It'll give you a full GenBank table, genome quality metrics, gives you what the machine learning model said, some summary results. It's a ton of information that you can use for basically anything. And that's all I have. So 35 minutes, I went a little bit over, but I want to thank everybody in my lab who helped me out with this. My committee was super helpful because I didn't come into this with a ton of coding experience. And I have the link to the GitHub page there. And I also want to thank CyVerse for letting me put Vibrant up on their platform. It's under maintenance right now, but hopefully it'll be ready soon. And I'll answer any questions that you have. Thank you.
Chris, thank you very much. That was great. We do have some questions, so we'll just start at the top. Here's a question. In my experience, as additional sequences are identified, they can be used as search templates for unknowns. Does Vibrant, your tool, have a link to updated data sources?
It doesn't. So if you're familiar with VirSorter, I believe it does have an externally uploadable database that you can put in. But that's mostly for applications like BLAST searching. So basically pairwise identity. So if you find a specific protein, you can see if there's something else in your database that has that protein, basically. But what Vibrant does is it uses HMMs, so hidden Markov models, and HMM search to look at profiles of proteins and the number of those proteins and what they mean. And it doesn't necessarily use pairwise identity homology. So uploading an external database isn't necessarily going to work because Vibrant would require you to cluster those proteins, identify the annotation for those proteins, give it a V-score, and so on. And what I've done is pre-built all of that before Vibrant went in. I can update the databases, such as KEGG and VOG have been updated since I published Vibrant. I think PFAM might be coming out with a new version soon. But you can see all the different versions. You can check out the GitHub page. It has which versions of everything I use. But that's my long answer to saying I don't think that feature isn't, and I don't think it will be available.
Okay, thank you. Another question is, could you please repeat what you said about the four ORFs that Vibrant can identify?
Yeah, so uh, that ORF stand for open reading frames, which is just a fancy word for proteins. So Vibrant, since it uses protein annotations to identify viruses, if the sequence only has one protein, Vibrant can't really tell if that is a virus or not because there's no contextual information. So in order for Vibrant to make a prediction and have enough information to not have high rates of false positive identifications, it needs at least 4 proteins on the scaffold. So 4 is the minimum set limit. And if you want to only analyze viruses that are of high quality, encode a lot of proteins, or if you want Vibrant to run faster, you can set that number to like 6 or 10, and all of your predictions will have 6 or 10 proteins on them. Same thing with the nucleotide cutoff. It's set to 1,000, but you can increase that if you want to.
Okay, somebody asked about your GitHub link, so we'll make sure that that is also on our website page for your webinar. And the next question is, can Vibrant be integrated into commercial packages like Omics Box?
Really quick about the previous thing. The GitHub link is here if you want to try to just copy it down from the PDF that gets uploaded.
It's also on the chat window too. So.
Okay.
Mm-hmm.
So can Vibrant be implemented into other pipelines? I don't know what Omics X is. If you are using a commercial program or commercial software that maybe you input your FASTQ reads and the software will trim your reads, assemble your data, figure out the best assembly, and then give you that assembly, then yeah, Vibrant can easily be implemented into that. I guess it depends on the application. Vibrant is just a Python script. That's all it is. In a very reduced form, it's just Python. So anything that can input a Python script will work just fine. And there's really not that many dependencies. So I don't think there should be compatibility issues.
Okay, somebody asked about sharing the slides. Again, as I mentioned, we do include a link to slides on the events page for this webinar, so that will be up shortly later today. And then another question, why use Vibrant on CyVerse rather than on the command line?
So Vibrant works just fine on command line, and personally, I don't use CyVerse for Vibrant, and that's because I mentioned that we have a nice lab server, but that's a $50,000 Linux server, and obviously not every lab has that. So if you have access to computing resources of any kind, then Vibrant is built very nicely for the command line since it's just a Python script. If you're analyzing a small data set, then it works again just fine on your laptop. But if you don't have access to those resources, or if you don't have access to storage, because CyVerse has free storage options, then uploading your data to CyVerse and using CyVerse's computational power is the main reason why that can be beneficial.
I'll just add my shameless plug as well, in that, why CyVerse was built was for doing everything in the same place. So ease of accessibility to your data, uploading your data, sharing your analyses, visualizing the whole thing. So as Chris said, if you have the command line skills, by all means, more power to you. But for those who want to explore maybe what Vibrant can do on some of their data without incurring costs for buying new Linux servers, give CyVerse a try and use Vibrant on it. Next question, how to identify the free viruses and pro viruses. I think the question is, does Vibrant identify the free viruses and proviruses, and how would you see that on Vibrant?
So yeah, I was just gonna quickly just put this here. That's all you need to run Vibrant. So that's the minimal command line. So if you're interested, I think everybody can see that. That's all you would need. So that's for command line. So the question, answering the question, if you look at the bottom left here, in all these different files that Vibrant outputs, there's a folder called Vibrant Phages, and Mixed Examples is the name of the file I put in. You'll see that there's files for combined, lysogenic, and lytic. So combined will have everything. That's where your GenBank file is too. But lysogenic are all the predicted integrated viruses or can integrate viruses, like viruses that are identified with integrase or within a bacterial scaffold. And then lytic is the free ones. There's also on the right a file called genome quality. So that'll tell you if it's high, medium, low, or complete genome quality. But that also tells you if it's lytic or lysogenic. And if the scaffold was excised or cut out of a bacterial scaffold, if the virus was cut out of a bacterial scaffold, then there'll be a name, there'll be a term called fragment in the name of the virus to designate that that fragment was excised from a bacterial scaffold.
Okay, thank you. And the final question, why can't you apply this pipeline / protocol to metagenomes from eukaryotes?
Right, yeah, so I can answer other questions related to that, but the main reason is because viruses infect prokaryotes and viruses infect eukaryotes are actually very different. They're similar in some ways, but there's really not a way to build them together in the same program. So that's the main reason. The other reason is because I built Vibrant to run as fast as possible. So I don't want to just throw all the possible computing resources and just do everything possible. I wanted to streamline Vibrant to work quickly as possible. So to do that, I took out most of the eukaryotic annotations. So any CAG or VOG annotation that should annotate a eukaryotic virus or eukaryote organism, I did my best to remove those. So that's another reason why.
Okay, I'm going to skip one question and come back to it, but the follow-up question to that could you recommend any similar programs, I guess, to Vibrant that work for eukaryotic virus catching?
I haven't looked hard into what's available, but I also haven't specifically seen any programs for that. I believe VirFinder has, it doesn't use any annotation, it just looks at nucleotide patterns, which have their positives and negatives. But I believe VirFinder can or has been trained on eukaryotic viruses. I would really look into that before you try that out though. But I think the best method I've seen people use is just take the eukaryotic viruses from a database like NCBI and then run a homology blast search. And then you'd have to manually look through that. I don't think there's really any good tools. And I think that's also because eukaryotic viruses share less similarity than prokaryotic viruses between each other. But I don't have a good suggestion, really.
That'll be a project for some future doctoral student, right? Okay, one last question. After downloading the Vibrant databases using the download-db script, if I want to move those databases to a different directory due to memory constraints, can I move the entire databases directory?
I think so. So what that question is referring to is after downloading Vibrant by a Bioconda install, I didn't personally build the Bioconda install, so I'm not exactly sure, but I believe the answer is yes. You can take that whole folder and move it somewhere else. What you would have to do though is Vibrant has a additional flag called -D, I think it's -D, and this will tell you where are the databases. So if you have it in your users' databases, you would give that flag to designate where you've moved that folder to. There's also one for the files folder. So I believe that should work, but I can't say that I know that that will work if you do the GitHub install. You can move the databases wherever you want with a GitHub install, and then just use that -D flag from then on. But again, it should work with Bioconda, but I'm not entirely sure.
Okay. People are thanking you, but if there are any other questions, type them in there. Otherwise, I'll just give you a quick preview of what we'll be presenting in 2 weeks on March 13th. Our next Focus Forum webinar will feature David LeBauer, who'll give us some solid grounding in TerraRef, a publicly available USDA-funded database of crop sensor data. He'll show us how we can access the data for our own research. It's an incredible database. It involves, remote sensing and how to make your metadata more, improve the fairness of the data, and all kinds of sort of how to deal with huge data sets. So please join us for that. Go to our events page and you'll find the webinar information. Thank you so much, Chris, and everybody. We'll hopefully see you in 2 weeks.
And thank you for listening.